Since I was little, I always wanted to be like David Attenborough. I was really interested in finding out how life works, so I studied biology and specialized in genetics to be able to do this. After we figured out that the genetic information was encoded in the DNA and then the structure of the DNA was solved, the next piece of the puzzle that was missing was figuring out what was the message encoded in it. Our genomes are composed of nucleotides represented by the letters A, C, T, and G, and sequencing is finding the exact order in which these letters are arranged. So during the decade of the 70s, there were several techniques developed for doing this. One of the most popular ones was developed by Fred Sanger, and this technique allowed us to read several hundred nucleotides at a time. During the 90s, the first Human Genome Project started, and it was finished in 2003 with the first draft of the Human Genome Project, and this was sequenced using Sanger sequencing technologies. After this, there was a craving to have more human genomes sequenced, so the next generation sequencing technologies started. With these technologies, like the ones that we have at the Crick, we are able to sequence over 50 human genomes in 2 days and at a fraction of a price. One of the biggest challenges nowadays is to reduce the cost of sequencing and also to find out what the message that is encoded actually means.